Can everyone hear me? Am I too close? Too far from the microphone? Is that good? Okay. So good morning. My name is Paul Siegel. I'm grateful to Dr. Zellner for inviting me to be part of this panel. It's great to be talking not just about psychoanalytic research, but specifically part of a panel devoted to more basic scientific research. The overwhelming majority of psychoanalytic research is about psychoanalytic treatment and showing that it works, and of course it's very important. But scientific revolutions aren't usually caused by clinical research. And the research that's been presented this morning, just sort of more basic, while certainly clinical implications and phenomenology, is really trying to get at basic psychoanalytic ideas. And it's really great to be part of a panel about that. Now, my research is even more basic than what my co-presenters have been talking about. Sort of a difficult act to follow something as clinically rich as borderline personality disorder to come and talk about phobias. But I'm going to be talking about why I think that's extremely psychoanalytically relevant. This is my research question. And I want to start also by saying that this is the kind of a question that would only be framed, as I'll get into, by someone who thinks psychoanalytically. It really cuts against the prevailing zeitgeist of cognitive behaviorism, in which the established dogma is that in order to reduce fear, one must directly confront their feared object. That is a dogma that's practically drilled into the head of every clinical psychologist. And the paradigm I'm going to be describing for studying this question is a neuropsychoanalytic one, because it's an implication of both psychoanalytic theory and contemporary neuroscience. I want to, before I get to the paradigm itself and the research, a brief vignette. An 8-year-old boy, a former patient of mine, whose parents were engaged in a pretty ugly divorce, he would create these epic battles from military history in my office, down to the last detail. And after a recent fight with the boy's father, the mother began a child guidance session. She sat down, she looked at me and said, "I hate drunks." And I thought to myself, well, that's interesting. Why did you marry 2 of them? And why are you currently dating someone who sounds like he's going to be the 3rd? Dynamic conflict is perhaps Freud's most enduring legacy. It's the cornerstone of all psychoanalytic models of the mind. And Freud believed that the phenomenon of the repetition compulsion was the clearest and most dramatic demonstration of dynamic conflict. Yet experimental demonstrations of dynamic conflict are exceedingly rare. It's not an easy thing to do. It's an unconscious phenomenon. It's very difficult to demonstrate the null sensitivity of any measure of awareness. And the phenomenon itself is dynamic. It involves the interaction of multiple variables. The predominant approach to demonstrating dynamic conflict has been to operationalize it a priori, to adopt a standard definition of dynamic conflict and that's based on observable variables, and then measure those variables. And without giving examples, I'll just say that this approach has not been particularly successful, has not changed thinking. And if you read Kuhn's The Structure of Scientific Revolutions, it's no wonder, because what experiments that have shifted paradigms, what they've all had in common is that they've used the methods and the techniques of the prevailing paradigm in novel ways. They've used them to study conditions that the prevailing paradigm had ignored, and thereby demonstrate new phenomena. I believe that the field of neuroscience has been approaching demonstrations of dynamic conflict, albeit unwittingly, as I'll get into. Over the past 2 decades, there has been an explosion of interest in the unconscious origins of fear. And as I say, as I'll get into, there's been, I think, unwittingly approaching demonstrations of dynamic conflict. And this paradigm, in a nutshell, is an attempt to bring forth that phenomenon, to make the unconscious conscious, if you will. First, some background. Research on unconscious processes is primarily based, especially in the scanner, is based on a technique called backwards masking. There's a fixation point, the X. This is very slowed down, so you can see what the target stimulus is, which is an image of a spider. And then there's a second masking stimulus, the rows of capital letters. In my lab, we find that the letters A, B, C, D work exceedingly well as a mask. And if the target is up there for a short enough duration, typically less than 30 milliseconds or 0.03 seconds, the participant, even phobic participants, are unable to detect the target stimulus. And this paradigm has been used to demonstrate a number of unconscious fear phenomena. It's been used to show, without getting into the details of studies, Arnie Oman in particular has shown that fear can be activated in the body entirely outside of awareness, can even be learned, can be conditioned outside of awareness in normal participants. You could take people who don't fear a stimulus and condition them with this masking paradigm by pairing the target stimulus with a shock, and fear could be acquired unconsciously. In a similar study, he also showed that the emotional experience of fear can be unconsciously evoked using this paradigm. And finally, neuroanatomical studies have showed clear dissociations between what people learn in the process of learning to fear something. There's a dissociation between what they consciously, cognitively know and their actual emotional responses. Now, it was Joseph Ledoux who established a biological basis for such phenomena in his discovery of dual fear circuits in the rat brain, what he calls the low road and the high road. He actually gave the Brill lecture in this room in 1997. His book, The Emotional Brain, had just been published, and this was what he presented. By progressively lesioning rat brains, he showed that there are two basic pathways of fear. The first, what he calls the low road, it's literally lower in the brain. A signal is a threat signal sent directly from the thalamus to the amygdala, which is nearby, not that many synaptic connections. Simultaneously, signal is sent up to the cortex, many more synaptic connections. It's longer, it's slower, but presumably permits, at least in his theory, for a more thorough evaluation, appraisal of the stimulus, what we commonly call perception. The low road, he proposed, is totally unconscious, occurs outside of awareness, initiating physiological responses in the body. Neuroimaging studies, I don't have time to go into the details about them. While our neuroimaging technology has not advanced to the point that we've been able to identify the circuits in the specific detail that Ledoux has neuroanatomically, they have similarly robustly pointed to a dual architecture of fear. There have been over 100 studies that have shown dissociations between conscious and unconscious processing of fear-relevant stimuli. Now, when Ledoux gave his talk here, and he gave the Brill lecture in 1997, someone in the audience, the first person, asked the obvious question, which was, what are the implications of this for psychoanalysis? And the audience was understandably underwhelmed by his response, because he nicely, tactfully said that he didn't see any, with busts of Freud standing on the other side of him. For me, that's probably a good question. Probably a dynamic conflict and compromise formation. Actually, no, it's not a defensive response, I think. Anyway, for me, and I thought this at the time, the existence of dual fear circuits to me raises rather obvious and interesting questions about what happens when the reactions generated by these circuits conflict, as in the phenomenon of counterphobia. Like any other phobic, a counterphobic experiences fear when they encounter their feared object, but they are interestingly able to override the fear response, override the avoidance of the object that is ordinarily elicited by the fear response, and instead they approach rather than avoid the object. In a few minutes, I'll get into how Brad Peterson, who I'm grateful to be collaborating with me on an fMRI study, he's the director of MRI research at the New York State Psychiatric Institute, how Dr. Peterson and I are using counterphobia in an attempt to demonstrate dynamic conflict neurobiologically. So based on all this prior research, my former mentor, Dr. Joel Weinberg, and I developed a paradigm known as very brief, or what I can actually call it here what I want to, which is unconscious exposure. I can't use that term when I write papers because no one will publish them. Can't get articles in major journals if I do that. As a matter of definition, and this is based on the backward masking paradigm, the participant sits in front of a computer screen or in the scanner, and they're bombarded by these masked images. As I said before, they're coming much faster and more furious. They get a bunch of them at the time. Each of them is for less than 30 milliseconds. And awareness tests demonstrate that participants lack awareness of the stimuli, both subjectively and objectively. Now, more importantly, this particular paradigm, when there's continuous exposure to the phobic stimulus over and over and over again, this is based on, our idea for this comes from psychoanalytic theory of phobias and trauma. The notion that inside of every phobic there is a counterphobic, right? An unconscious wish to master what one fears. So that despite his motivation to avoid some dreaded scenario, a counterphobic nonetheless finds themselves again and again knee-deep in this scenario. If counterphobia is indeed underlied by an unconscious wish to master fear, then when phobic stimuli are presented unconsciously under conditions that prevent the activation of defense mechanisms, this should fulfill the unconscious wish and thereby reduce conflict. Right? That's the theory behind it. And if psychoanalytic theory is correct, the phobic person should be made less resistant or avoidant when they subsequently approach their feared object. And in an initial study, that is exactly what we demonstrated, in which arachnophobia participants were approached a live tarantula significantly more. There were clinically as well as statistically significant differences, much more than after they were exposed to the same stimuli unmasked or clearly visible. And I want to point out that psychoanalytic theory, again, uniquely makes this prediction that, that unconscious exposure to feared stimuli should reduce fear, should promote approach towards a feared object. It directly contradicts the dogma of direct confrontation of cognitive behaviorism. So in very brief exposure, one thing we're trying to do is directly pit the opposing predictions of psychoanalytic theory and cognitive behavioral theory against each other. Now, the more recent study, I'm first going to be talking about the behavioral study that we did to correlate with what we're going to do in the scanner. As you know, it's difficult to study behavior in the scanner. You want to study brain-behavior relationships, but people can't move. So our strategy, and we want to study things like phobic avoidance. So we first did a behavioral study in my lab at Purchase, comparing the effects of conscious and unconscious exposure to phobic stimuli. And I'm pleased to be able to say I recently found out this study is going to be published in the journal Emotion, which is a flagship journal of the American Psychological Association. I was going to say Psychoanalytic Association. And this study I'm grateful to the Fund for Psychoanalytic Research of the American Psychoanalytic Association, who gave me all the money to do this study, and allowed us to get a sample of 101 arachnophobic participants and pay them and actually get this study done. This study would not be possible without their help. To identify our participants, they're all undergraduate students at Purchase, enrolled in taking social and natural science courses. And they fill out a questionnaire called the Fear of Spiders questionnaire. It's kind of a quick and dirty measure of whether or not they're afraid of spiders. That's how we identify people. But we, of course, don't take their word for it. When they come in the first week, the first thing we do is we put them in a room with a live caged tarantula, who I've nicknamed Fuzzy to deal with my own phobia or counterphobia. And they do something called the behavioral avoidance test, in which they have to gradually approach Fuzzy. Those are two of my research assistants demonstrating the BAT. It's an 11-point scale of willingness to approach Fuzzy. If the participant is not even willing to enter the room, and that usually doesn't happen, their score is 0. And all the way up to the final task where they're willing to actually put their hand in and touch Fuzzy. We don't let them do that. They have to do all the other tasks. We don't let them touch Fuzzy. Fuzzy could bite. And if they're allergic to tarantula venom, that could be really bad. But the important thing is that if they say they are willing to touch Fuzzy, they think they're going to have to do it because they had to do all the other tasks up to that point, so they have a response set at that point. So this is, we then confirm that they are in fact phobic. The week 2 is the second, the heart of the experiment, where we did our actual manipulation. just prior to the manipulation, we got a measure of how afraid people were going into the experiment by asking them for what's called a subjective units of distress, or SUDS rating. It's basically telling someone how afraid you are on a scale from 0 to 10. As simple and rough as that sounds, it's actually the most widely used measure of fear. And then we did the actual manipulation where participants were continuously exposed. They got very brief exposure to either spiders or flowers. It's a between subjects design, so people were in one of these three groups. Flower is a perfect control stimulus because like a spider, it has a central body with protrusions, multiple protrusions. Like a spider, it's alive. But unlike a spider, it is not a biologically prepared fear-relevant stimulus. So that's our control stimulus. And then the spiders are also presented unmasked, that is clearly visible on the screen, right? So the main thing we're manipulating here is awareness of the stimuli, right? Which is basic to any study that's looking at unconscious effects. Immediately after the manipulation, the first thing the participant does is they give another SUDS rating to see if their subjective experience has changed as a result of the exposure. And then we have them approach fuzzy again to see if there are changes in their phobic behavior. Finally, there's a funneled interview to assess their knowledge of the stimuli. 2 weeks later, 2/3 of the participants, I did mention before about it, there were 101 undergraduate students. And 2/3 of them returned 2 weeks later to do the BAT once again so we could see to what extent the effect was maintained. So rather than showing you means, a picture says 1,000 words, and I don't have that much time. So you see the three types of exposure and their effect on the BAT. This is our primary dependent measure. What we care the most about is how it's actually affecting their ability to approach fuzzy. And as you can see, the very brief exposure to spiders shown in blue, the masked spiders, you see this big increase from the baseline to post-manipulation. That turns out to be, again, not just statistically significant, different than control. It's also significantly different than exposure to the same stimuli within conscious awareness, the unmasked spiders. And the effect size was pretty big, about 0.4, which is respectable for a kind of basic behavioral study. So the masked spiders appear to be affecting behavior. Keep in mind, I should have mentioned this before, the higher the BAT score, the less fearful they are, right? And the more willing they are to approach fuzzy, right? So you see an increase in their willingness to approach the spider. So that's the behavioral effect. Now effects on their conscious experience of fear, on the SUDS measure, subjective distress, and you see there's exactly the opposite pattern. Now there isn't much of an effect on the masked spiders, right? Or it's paralleling basic control. The baseline measures are somewhat different, but not significantly so. But now we see that conscious exposure, this is actually a pretty mundane finding. When people see spiders, consciously they get afraid. That's all this finding is showing. But what's interesting is that it's exactly the opposite of the behavioral finding, right? So that we see that there's what we call a double dissociation between the effects of the conscious and unconscious exposure on behavior versus subjective experience, right? Unconscious exposure affected phobic behavior. In fact, it reduced it, but did not affect their conscious experience of fear, whereas conscious exposure had exactly the opposite effect. It didn't affect their phobic behavior, and it increased their subjective experience. The reason why this double dissociation is important is it suggests that the effects took place outside of awareness, right? And so you had an awareness manipulation, and that awareness manipulation, you had these dissociated effects between behavior and consciousness. This study was, and this double dissociation specifically, was just replicated in my lab with another sample of 88 arachnophobic participants. So, the first time you get it, you think maybe you got lucky. The second time, you're like, well, this really might be onto something. And then the third time, you may become a true believer, and that's very dangerous as a scientist. The other thing the study demonstrates, as I say, is that, in a sense, less is more. At less exposure to unconscious stimuli reduced phobic avoidance more than exposure to the same stimuli within conscious awareness. And again, this is exactly the opposite of what cognitive behavioral theory predicts. It refutes the exposure dogma and is consistent with psychoanalytic theory. The other thing that happened is that I said that we measured the effect 2 weeks later, and the behavioral effect was in fact maintained, which also runs counter to prevailing cognitive theory about the effects of subliminal stimuli, which are believed to, fade very, very soon, minutes after a manipulation, and we found them 2 weeks later. Probably because we're looking at unconscious affective stimuli, whereas most studies that have looked at the effects of subliminal stimuli have used stimuli that are more cognitively rather than affectively mediated. The theoretical implications, these data suggest that there was unconscious habituation to the stimuli, that by being repeatedly exposed to the feared stimulus, right, eventually you got decreased responding to it. That may sound like a behaviorist interpretation. It's important to emphasize that habituation is not a construct of behaviorism. Habituation is a ubiquitous phenomenon. It has been evidenced at every level of organismic organization in which it's been studied. Multicellular organisms, believe it or not, habituate to stimulation. It has not been demonstrated with respect to unconscious emotion. Right? And these data suggest that it happened. And if it did, the question this study raises is, how did it occur? What's the mechanism? Phobics are motivated to avoid what they fear. So why did they approach as a result of the manipulation? It likely is not due to changes in conscious processes because SUD scores didn't change. I believe it's because an unconscious wish was fulfilled and dynamic conflict was reduced. But what does that have to do with the price of eggs? Because this study in and of itself doesn't demonstrate it, of course. Right? This study is not, it suggests it as one possible explanation. But that's the point of the fMRI study. We're now just about to start. We've rounded up all our participants. We just need to figure out a way to pay them. And we have the paradigm set, the design set. We're now adopting very brief exposure to the scanner to link the behavioral effects with what's going on in the brain. And we're going to be comparing patterns of activation, again, of those 3 types of exposure. And we're using what's called a multiple block design, where people are going to get, like many blocks of these target stimuli. And we're doing that specifically to test the hypothesis of habituation by seeing if there's habituation of amygdala response. More importantly, the more important part of the operationalization as far as dynamic conflict is concerned is the inclusion of a counterphobic sample. We're going to be comparing phobics and counterphobics. As I mentioned earlier, counterphobics experience the same fear that other phobics do when they encounter fuzzy. We actually see this in my lab. Interestingly, about 1 out of every 5 of the phobics we identify as counterphobic. Right? And we have to exclude them. They go all the way to step 10. They're willing to touch fuzzy at the baseline. So they approach fuzzy as much as non-phobics do. Again, there's this dissociation between their behavior and how afraid that they say they are. And interestingly, they are usually not aware of it. Reminiscent of the repetition compulsion, when you ask them, you say you're afraid, but you were willing to touch fuzzy. And the participant will typically say, well, you asked me, so I said yes. Right? So presumably in counterphobia, brain circuits associated with the motive to master fear go online. And if that is the case, a likely candidate is a circuit that projects from the amygdala to the ventral medial prefrontal cortex. I don't have an image to show you, but it's right here. This region of the brain was made famous by Antonio Damasio, who showed, again, neuroanatomically that this region of the brain is central to regulating emotion and emotional reactions. The ventral medial prefrontal cortex has dense connections with the amygdala and the limbic system. fMRI studies of fear regulation have consistently pointed to this region of the brain. And in studies of fear learning, including studies of mass stimuli, what you typically see is you have amygdala activation and then followed by ventromedial prefrontal cortex activation, demonstrating, I don't have time to go into the details, but demonstrating a possible dynamic relationship between the activation and regulation of fear. And what we're going to try to do, again, is to sort of to bring that forth by comparing phobics and counterphobics and the effects of conscious and unconscious exposure on them. Now, with conscious exposure, which are the unmasked spiders, this is just going to confirm what other studies have showed. We're probably going to see, we think, amygdala activation followed by the ventromedial prefrontal cortex activation, with the ventromedial prefrontal cortex activation kind of lagging behind amygdala activation. With the very brief exposure in the masked spiders, we believe will be curvilinear amygdala activation. That is, over blocks. There'll be many blocks. There's going to be 3 runs for each of the 3 types of exposure. Each of them is about 10 minutes, right? So they're going to get like 400 of these stimuli. And over the course of blocks, time series analyses, we believe, will show that amygdala activation will first, spike, and then it will decrease, showing a possible habituation effect. At the same time, we think the multiple blocks of stimuli eventually will start to increase arousal, so that we'll also see ventromedial prefrontal cortex activation. So that this is basically a way of saying there's kind of a beginning of a dynamic phenomenon. And then with the counterphobics, this is where we believe the dynamic phenomenon will more fully emerge. Again, with looking at the mass spiders at the bottom, where again we'll see curvilinear amygdala activation, sort of an increase and then falling off. And we believe that the ventromedial prefrontal cortex will show a similar trajectory, lagging behind the amygdala, but we'll also start to see habituation there. Again, that because of the counterphobic wish to master fear, right? After you get enough amygdala activation, because of the counterphobic wish to master fear, the ventromedial prefrontal cortex will be activated sooner, right? And thereby have more of an opportunity to habituate across these multiple blocks of stimuli. So that taken together, the activation trajectories of inphobics and counterphobics of very brief versus conscious exposure, we'll see a dynamic relationship between the unconscious activation and regulation of fear, or dynamic conflict.
Can someone who's back by the lights turn the lights on for me, please? Thank you. So we'll take a couple of questions for Paul's talk, and then we'll have open questions for any of the panelists.
I gather you see the masked exposure as an aspect of that could be used in therapy and reducing the avoidance. But a behavior therapist might say, well, that's all very good, but in behavior therapy, we use the unmasked exposure, and then we train them behaviorally with relaxation and all of that. And then they end up both unavoidant and unafraid.
Right. Yes. The response to that is that we know from epidemiological studies that precisely because it involves direct confrontation, that the overwhelming majority of phobics actually do not go to treatment because they know that they're going to have to face what they fear. So yes, it works very well. Behavior therapy works very well, those who are willing to accept the distress associated with it.
I just have a question about the spiders, which I think I'm a little bit kind of counterphobic to the spiders. Mm-hmm. So when you show the tarantula, in my mind, I would go right up to the cage. But I would definitely stop when it said touch, and I'd say, I have 2 questions. Does it bite? Is it venomous? Will it hurt me? And unless I got absolute reassurance, no, I wouldn't touch it. But if they said yes, I would, with a lot of, certain counterphobic.
Right.
Do any of your design subjects, when they go up to it, stop and ask, is it going to bite me if I touch it?
Yes, they do. And we can't answer them because, of course, then we'd be reassuring their anxiety, and that would introduce a major confound into the manipulation. So we don't do that, but they do ask. Before they enter the room with Fuzzy, they read a sheet describing all of the tasks that they're going to do, and they are told at no point will you be in any danger. That you have to say that. So that's sort of ethical, indirect reassurance. But when you put them with the actual room with the spider, they tend to forget about that and do exactly as you describe.
So while questions are percolating, I have one that's sort of, I suppose I should turn this off. It's going to be loud for a minute, but So I wanted to ask any of you guys sort of what about having a psychoanalytic approach or understanding is it uniquely different than the other kinds of literatures that are involved sort of with looking at your particular research question. So 2 examples would be that, Luis, you had talked about that the alexithymia, that the lower level of emotional awareness could actually be an outcome of a defensive process. So I'm assuming that that would be different from sort of other modalities. They would just think of that as sort of a primary deficit as opposed to there's a capability that's being suppressed. And then of course Paul, your take on phobia has a very dynamic aspect in terms of the counter wish to approach. So I'm just wondering if you guys can share a little bit more about what do you feel is specifically different about a psychoanalytic take on your research question, and then also if you've encountered a lot of hostility or resistance by sort of integrating that aspect into your research. And you guys can just share that mic there.
Yeah, I mean, I think the example that you raised in terms for me, actually that data set had already been collected when I started working with the group I'm working with, and I was telling Jeff actually that the person I was working under didn't know how to interpret the data. And so she sort of gave me this pattern of data with alexithymia and high alexithymic borderlines, low alexithymic controls. And it seemed fairly obvious to me, actually, that at least that might be a possibility. Now, it's not proof, obviously, but the other thing that I would mention is that I think, as someone in the audience already intimated, that all of these kind of functional MRI studies, you get this sort of blobology of brain regions, and it's hard to know what to do with that information unless you have a guiding theoretical approach. And I mean, I think you see that a lot of times with reductionistic approaches where, this brain region is involved in this, and then you'll read another paper and see that this exact same brain region is involved in a totally different process. I think psychoanalytic theory helps to bridge those gaps to some extent.
Well, I think, considering the paradigm that I'm using, I think the measures are highly influenced by psychoanalytic thinking, and there really aren't a lot of alternatives for looking at these sort of constructs, because the things that we're attempting to measure are really based on mental representation phenomena. So thinking about object relations and a very in-depth dynamic view of the relationship with the parents, I think. I'm not sure what the alternative would be for that. So to my mind, what I'm trying to do in my work is to bring that component in to what is otherwise, I think, a more, maybe a more restricted dialogue.
I had a little trouble with your question for a second because I actually presented some of this work at the World Congress for Behavioral and Cognitive Therapy over the summer on a panel of mostly cognitive behavior therapists and experimental psychopathology researchers. I actually did not explicitly use too many psychoanalytic constructs or jargon or anything like that, and nobody really had any problems with any. It's not like any of the findings themselves were that shocking or unpalatable or uninteresting to them. So in a way, I wonder if that's even a question we need to be worrying too much about. But at the same time, I do think there are some implications. The thing that I tend to think about, and maybe this is because it's something that I do, are that the data I presented suggests the importance of looking at transferential phenomena in treatment. And that if you, for example, just assume that people with borderline personality just sort of have a problem recognizing social cognition and setting up some sort of, maybe setting up some sort of cognitive behavioral homework around that or something, you might miss the very live, real, current, in the room phenomena that could be highly therapeutic if, with proper training and technique to help them change and reevaluate and modify the way in which they trust or don't trust you as a therapist. So I think psychoanalytic treatments are very uniquely positioned to be able to think about that in a very sophisticated way that other treatments haven't.
I would answer your question by saying simply that I've been taught to think differently about, so that if I will look at a phenomenon like exposure or habituation or, cognitive behavioral people who do both basic and clinical research, they have an easier time because they tend to study linear relationships and linear phenomena, which are more easily adapted to experiments. But I've been trained to think by thinking dynamically. I'm thinking in terms of multiple variables that are interacting, and I try to think about how in an experiment to create conditions that will show that. So basically, not just to think dynamically about things. As far as the bias, it's interesting. When I present my stuff to neuroscientists, it's very different than when I present. And they don't have to be psychoanalysts; just dyed-in-the-wool neuroscientists. They have no trouble believing it. They say, "Oh, no one's done this? How could it be otherwise?" When I present the same stuff at clinical conferences and cognitive behavior one specifically, people get very upset. People don't believe me. They think I'm presenting apocryphal data. There's a lot of bias and a fair amount of hostility. I sometimes get hate email messages because it subverts the dominant paradigm.
I think it depends on which one has the most trustworthiness.
Okay, so I'll take some questions from the audience.
Hi. I have a question, realizing the limitations of feasible research, and I know it's easy to imagine things you'd like to do and harder to actually do them. But in terms of how deep is the bias toward untrustworthiness, I know there were some studies, and I don't remember the author, where they showed a little boy in a masked image holding a cake, smiling, or throwing the cake at you. And then they asked the person consciously with a picture of the boy what he thought of the, what attributes would you give this boy, who was basically neutral. And the ones who saw the masked image of the boy throwing the cake at you came up with more negative adjectives. So just wondered if you had your controls and told them that some of the people they were going to see in the faces were actually criminals. Could you introduce a bias, and if so, would it duplicate the personality of the effect from the borderline personality? Because I think it's very hard to distinguish sort of transient perception problem from a deeper underlying problem.
Yeah, no, that's a fascinating thing to consider. I'm not familiar with that particular study, but there are studies that do things like that, where they'll show a face and tell a story about that person as an attempt to manipulate whether they're kind of like a friendly person or an unfriendly person, or trustworthy or untrustworthy, and then use that to look at both subjective experience parameters, but then psychophysiological or physiological parameters like galvanic skin response to these faces and whatever. So I think that would be one of the directions I'd like to go with this, is to what degree can you change or manipulate trustworthiness based on contextualizing features, and then in relevance to treatment, like, can you change it once it's established, which I think a lot of people with borderline have trouble doing.
I would just add one thing to that that I think Eric is implying. I think the paradigm that we're working with the trust appraisal is a relatively new one, and looking at trait appraisal is something that has a lot of potential in terms of being able to develop it and look at different contexts within which people are trusting. So I think both your question and the response speak to this, that there's a long way to go, but we're just interested right now to find, what is the preliminary evidence. Yep.
Hi, this is a question about the phobia study. I was wondering if the participants that did show sort of less phobic response after being exposed to the masked images, whether they actually noted whether they noted the difference and were surprised by that. And I mean, because that's, you know, I know you said that the subjective experience was that they still had the same fear, but I wonder if there was, you know, I mean, implications for like treatment in terms of like actually sort of bringing that back to the conscious level.
Yes. Thank you for asking that question. It gives me the opportunity to talk about something I wish I had time to when I was presenting. The majority of people, no. There doesn't seem to be, there wasn't any awareness of that they got closer to fuzzy. They didn't say this, they thought it was because they were doing it the second time. But, you know, clinical data in the context of an experiment, by which you know, with particular participants can be interesting. So there were 3 participants who were struck and appeared to have insight into the process that something had changed. And one person who went up like 4 points. And interestingly, she had this ability to sort of become absorbed in the stimuli, which I think is a variable that may be mediating this effect, the extent to which they can kind of lose themselves and become absorbed in the images. She went up considerably, and she couldn't believe that she was doing this and commented on this and asked what had gone on in the room and if she could do it again. So that there were some people who seemed to be showing some kind of insight that their behavior was being changed.
Thank you very much for all the lectures, very good. I just was, to Eric, I just wondered simplistically if borderlines for the particular task, they just are recruiting the frontal brain, the orbital frontal brain, just because they need a little bit more oxygen to decide a neutral task. If not only because of that task, but many other tasks, they have difficulty deciding. So they have to recruit actually the frontal brain because that's one of the particular reasons they are recruiting more the frontal brain. And that's not only because of the trust or not trust, because that's yes or no. And then many other tasks, they have difficulty deciding. Particularly, so they have to recruit actually just the frontal brain.
Yeah, that's a serious possibility. That's why we had the fear condition as a comparison, and we did not see the same OFC differences when making a fear decision. So that goes some way to suggest that there might be something more specific, at least in trust or decisions related to trust, than at least another emotional decision that is presumably unrelated to trust. So at this point, that's all I can say, but at least in this data, this preliminary data, we're not, this maps onto the behavioral data too. You saw that the ratings and the reaction times were quite comparable when fear was more clearly being expressed in the face. So, that, in fact, this is the very reason we set up the experiment this way, so that we could rule out just a generalized attention or oxygenation difference of some sort.
So I think since we're going late, we'll just take one more question and then wrap it up for today.
This is about the spider study again. A couple questions. I wasn't sure, I couldn't visualize how people were having behavioral change but also expressing no change in their subjective experience. Mm-hmm. So they were walking but saying, I'm really feeling bad about this at the same time. So that,
yeah, I'm sorry for not being clear about that. So you understand the behavior was just the BAT, right?
Right. Mm-hmm.
So the way we measured, the SUDS measure, the subjective units of distress, mm-hmm. we just asked them to rate their level of fear on a scale from 0 to 10, 0 meaning no fear whatsoever, mm-hmm. 10 meaning being unbearable fear of the spider, 5 moderate but tolerable. Mm-hmm. They gave a number right before the manipulation, before they saw the images, and then directly afterwards. And that's the, as I said, actually the most widely used measure of fear, especially in cognitive behavioral studies and treatment studies and basic research studies, to get it, just because that's what people tend to care about clinically, is that the distress people are actually experiencing.
And so they would have the same, they'd have the same level of distress, but they would approach closer to the spider.
Correct. Correct. So the manipulation would not, and you have to do that right before and right after. You can't do that when they're starting to approach, because now the measurement is confounded with fuzzy, right? So you want to see, does the manipulation, you want to look at its effect on distress separate and apart from its effect on whether or not they approach fuzzy. So that's why we do the SUDS measure immediately before and immediately after the manipulation, and then they approach fuzzy again.
Okay. And one last question. I wasn't sure why the unmasked stimuli would be more effective than a masked. No, the masked stimuli was more effective than the unmasked.
Right, yeah, so you're saying...
I could see why that it would maybe have the same effect, that you would discover that, but that it was more effective. What's your understanding why it would be more effective?
Twofold. I think with as I said before, with the mass stimuli, I think that unconsciously there's some habituation going on, and I believe that is a dynamic phenomenon. For the unmasked, that I think is easier to interpret. That's a somewhat more linear phenomenon, where if they see pictures of spiders on the screen, right, that increases. They're now more distressed when they go in to see fuzzy, right? So that distress then interferes with their ability to approach an actual spider, right? So that's more mundane.
Okay, so I want to thank all of our panelists. I think that you can see that we're really hopefully entering, yet another chapter in our neuroscience and psychoanalysis dialogue, when we have, such great minds getting in there and really in the trenches generating some data. And we're going to have our next meeting on Saturday, December 4th, when we're having our speaker will be B.J. Casey. So again, thank you all to our presenters. Thank you.